Hi, I'm Jesse Major. I'm a professor at the University of Massachusetts in Amherst, Massachusetts. Um, my lab here is really focused on early mammalian development in the mouse. We have two major interests. Um, the first is in epigenetic events that regulate genomic imprinting throughout early development and adult life. And our second major focus is really about control of lineage decisions that happen during the earliest stages of development. So both pre-implantation and gastrulation, which is the two really stages that we focus on primarily. And my work has really dovetailed very nicely with the larger goals of the Knockout Mouse project and the IMPC in general. For many years, I've had also an interest in just functional annotation of the genome, that is, understanding what all genes do, mostly interested in how genes regulate early development. And in the past, I've done different phenotypic screens, all with this same purpose, to really understand all the genes required that regulate early development. We've interfaced really nicely with the Knockout Mouse project. We've developed a pipeline for assessing any early lethal alleles that arise from the production centers. And so currently we've gone through or working up to about 70 different homozygote lethal phenotypes that we've assessed, and we're still moving right along, working strong. And we really start during gastrulation. So we get only alleles that homozygous cannot be found at 9.5 by the production centers. And then we first assess gastrulation. We ask, can we find them at day 7.5? If we can, we ask if they're normal. We do some analysis there, both morphological and immunofluorescent analysis of lineage determination. If we cannot find them during gastrulation, we'll back up and try and ask if we can find these mutant embryos at day 3.5. And at that point, again, we do morphological analysis as well as some immunofluorescence to look at lineage determination in these mutant embryos to try and understand why each gene is resulting in a lethal phenotype and what's going wrong in these mutant embryos. That's sort of laid out in our pipeline overview. For each of these alleles that we're working with, at least many of them have a reporter LAC-Z knocked into the locus. So we're also performing just expression analysis on the LAC-Z generated or heterozygotes, the heterozygotes that contain the LAC-Z reporter, to simply ask where these genes are expressed during different stages of development, as you can see here. If we do find mutants, as I said, we embed them and section them and perform a variety of histological assays as well as immunofluorescence to ask if things like the primitive streak are forming normally, if you're getting all three lineages properly, ectoderm, mesoderm, endoderm, and so on. We're hoping to provide this kind of data back to the IMPC for each of the alleles that we're characterizing, as sort of, again, towards this goal of functional annotation of the entire genome. What's really fun, now that we are good ways into this project, is we can start to look at all the mutants we get together and really line them up next to each other and ask if there are trends that we can find during development. And this is not necessarily a novel idea. I mean, there are many other developmental biologists who believe that there are particular time points and developmental milestones that must be reached in order to progress. And this large-scale mutant analysis is really bearing that true. So we can clearly see that there are key transition points that we think if a certain lethal allele or lethal phenotype, the embryos cannot reach a certain mark, they simply stop progressing. And so we can see mutant phenotypes sort of piling up or looking very similar to each other at several different stages. This is true of the gastrulation phenotypes, where we see many alleles that make a nice egg cylinder, but it seems like they just stop growing and really fail to turn on or activate genes associated with primitive streak, which really is the hallmark of gastrulation. So these are really post-implantation, but pre-gastrulation phenotypes. So that's kind of cool. We have a large collection of these. The other sort of clear line is at implantation. So we have about equal numbers of genes where the phenotypes we can recover during gastrulation and those that we cannot. And of those that we cannot, almost all of them we do find at day 3.5, and they come in really 2 different flavors. Also, some mutant phenotypes that fail to make a blastocyst at day 3.5, and others where the blastocysts form perfectly fine, but they clearly have defects in the implantation process itself. And that's why we cannot find them 3 days later or 4 days later during gastrulation. So when we collect these early embryos, we perform morphological and outgrowth assays on them to simply ask, what can these cells do? Are they capable of hatching from the zona pellucida and forming outgrowths, which is required, sort of a required step during the implantation process. So that's sort of phase 1 of our sort of pipeline and analysis of early lethal mutants. What's also been great is that we're now able to pick genes that we think are particularly interesting and take a more in-depth view. And already in this work, we've sort of found several families of genes that we probably never would have picked otherwise to study. We have one large batch of sort of mitochondrial-related proteins or genes that all have similar phenotypes during gastrulation, which now we are following up on. Likewise, we have another family of genes, sort of a biochemical related metabolic pathways during pre-implantation that, again, we never really would have studied otherwise. We also have 1 or 2 that are connecting back to this first goal that I told you about, which is understanding epigenetic events during early embryogenesis. And we have some nice novel phenotypes that are arising from these newly generated lethal alleles by the by the Knockout Mouse project. And so this work will continue, and hopefully we'll get lots more interesting phenotypes and characterize all these early lethalities along the way.